This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Hello and welcome. Thank you so much, everyone, for joining us this evening for a brief parlay into the world of stem cell research here at the University of Melbourne and through our partner institutes at St Vincent's Hospital and at the Children's Medical Research Institute as well. I would like to start by acknowledging that we meet on the lands of the Wurundjeri people, and they've been custodians of this land for thousands of years, and I acknowledge and pay our respects to their elders past, present and emerging. So we're delighted to see you all here today, and I'm particularly delighted because aside from welcoming you, I'm able to hand over the floor to some really remarkable researchers who are part of a broad repertoire of stem cell research happening here at the University of Melbourne. Before I hand over to my colleague Megan Munsey, who is Deputy Director for the Centre for Stem Cell Systems, I would like to just point out a few housekeeping notes, and that is that we have bathrooms out to our left here. The emergency procedure, should we be asked to evacuate, is simply to exit the auditorium by the closest door and meet outside the Kenneth Myer building on the footpath. I am Professor Christine Wells. I'm the Director of the Centre for Stem Cell Systems. I've been here at the University of Melbourne for the last 3 years, and in that time been absolutely delighted to see a vibrant research community that encompasses not just excellent stem cell research and not just an interest and a drive towards translating that stem cell research, but also encompassing many of the other aspects needed for translation that include the investigation of the societal consequences of stem cell research, the legal and regulatory aspects, and of course using stem cells to understand our own biology and to apply this to a better understanding of development and disease. So you hear this term stem cell bandied around an awful lot, and before we get on and talk to some of our stem cell researchers, I just wanted to give you a very brief overview of what a stem cell is. Now, stem cells fall into two main camps. One camp are the cells that sit in your body, even as an old woman like myself, replenishing some of the tissues of our body, in particular tissues like our blood. And these are cells which are notoriously difficult to identify. They are, in the main, quite poorly characterised, and there is a section of, shall I say, our tourist industry, which relies on people believing that some of these cells, for example from your fat, have magical properties. These are not the cells that I will be talking to you about tonight, nor are they the cells that we will be exploring within our research component. The majority of cells, stem cells that we work on here at the Centre for Stem Cell Systems are pluripotent stem cells. These are cells which have genuine stem cell properties. They are able to give rise to many tissues and cells of the body in a laboratory dish, and some of those cells are worked on from animal models such as mouse or chick or zebrafish, but the majority of them are human stem cells, and they are derived from an induced pluripotent stem cell system. So my laboratory could take a piece of tissue from any of you sample of your blood or a sample of your skin and reprogram that tissue back into a cell that thinks it's in the early embryo. So it belongs to you, it has a memory of your genetics, it may even have a memory of some of the environmental events that you have seen during your life, and so that cell is uniquely able to model diseases that are unique to your genetic background. And using that cell, we can then mimic the tissues of heart, blood, kidney, brain, and so on. So today we are going to be talking particularly about the clinical research and community partnerships that make stem cells a really effective and promising model. So this is not about the false promise that might be marketed on a Google search of stem cell clinics in Australia, but rather about where we are on that path to translation, what the drivers are from community groups, and where we think we will be in terms of stem cell translation over the next few years. I'm really delighted that Associate Professor Megan Munsey is here with me as moderator. She has brought a very important aspect of stem cell research into the centre, and that is, of course, the aspect of the ethical, legal and societal components of stem cell research. But before Megan takes to the stage, it's my great pleasure to introduce you to our speakers. Here you can see that we have a number of researchers from the University of Melbourne partnering with key clinical and community partnerships. And first cab off the rank is Dr. Kiru Yap, who is a student at the University of Melbourne completing his PhD. Kiru is also a clinician, and I think he brings a really unique perspective as a clinical researcher who is also using stem cells to study disease. Thank you, Kiru.
Thank you, Christine. Thank you very much, Christine, and thank you very much for spending your time with us today. So I work at St Vincent's Institute at St Vincent's Hospital campus, and I'm obviously a clinician. And our group consists of a couple of people, including clinicians as well as laboratory scientists. And our work mostly revolves around the construction of mini liver tissues in the lab, which we call organoids. And organoids because they mostly resemble the structure and function of organs that are found in the body, in our case, the liver. Some of the unique things that our lab is doing includes making complex liver organoids that enable or incorporate additional structural elements in the liver, for example, blood vessels that enable the exchange of oxygen, excretion of carbon dioxide, as well as nutrients to come to the liver, as well as bile ducts, which in the liver itself usually enables the excretion of metabolic byproducts, such as bile. One of the other things that as a surgical group we are interested in is to make new novel ways to transplant organoids into the body. So new cutting-edge surgery that is not currently being done to transplant these constructs that we're making in the lab in order to treat liver disease. And this sort of research is critically important because in the developed nations we are currently facing a silent epidemic of liver disease. Partly because liver disease is associated with metabolic conditions like obesity and diabetes, which gets a lot of focus, and the liver is often an afterthought, but also because the liver affects a wide variety, there's a lot of exchanges with a variety of organs in the body. So if it affects the pancreas in diabetes, if you have Alzheimer's, for example, there's a liver component into it. If you have cardiovascular disease, there's a liver component to it. And the liver can be damaged in a lot of other variety of ways. For example, viral hepatitis, which is an infection. You can have alcohol as well as drug-induced liver injury, and you've got genetic liver diseases, just to name a few. But a lot of these diseases have the same underlying process where you get a lot of inflammation in the liver, which leads to scar formation. And this is often a silent process where patients are asymptomatic, so they don't have any symptoms. And when a patient finally turns up to us, it is often when they're almost at liver failure, and therefore they desperately need a liver transplant. Or a lot of these pathogenic processes leads to liver cancer because the cells mutate in that harsh environment. So a patient often turns up with a liver cancer, and then you find the liver disease that should have been treated many decades ago. So it is important that we come up with new ways to both diagnose but also to treat these patients, because a lot of liver disease at the end stages needs a liver transplant, but there's simply not enough livers to meet demand. So a lot of patients die before even leaving a transplant. So an innovative solution that's grounded in regenerative medicine really offers the potential to challenge the way that we treat these patients. So we generate liver organoids in the lab from broadly two different types of cells that Christine has alluded to. So on this side, you've got cells that have been derived from patient liver tissue. So patients who undergo surgery at St. Vincent's Hospital have kindly donated their tissues for us to use in research. And we can isolate important liver cells from this to enable us to construct a patient's organoid in the lab. And on this right-hand side, we've got cells that have been differentiated or matured from human-induced pluripotent stem cells. And those are the master stem cells that Christine just alluded to, where you no longer need to take a piece of liver of a patient to make new liver for them, but you can simply have a cheek swab, can donate some blood, or even urine, for example, make the iPS, and then bank them, expand them when you need it. And that means that I can make my own liver when I need it in unlimited quantities, on demand, and potentially transplant it back into my body. So we've developed a variety of different methods to generate these organoids in the laboratory, and I've got two on top shown over here. And basically what we do is we take cells, so the main cells in the liver as well as all the other liver cell types, either from human patient liver, or we derive them all from iPSC. We combine them together into we're using special techniques that we've developed to make these three-dimensional constructs. And on the bottom over here, I've got images of what happens when you transplant these organoids into the bodies of animal. And what basically happens is you've got the liver cells, or the main cells in the liver, forming hepatic epithelium. So those are the main structures within the liver, which are found over here. So that's in white, and that's in green over here. And surrounding all those cells are blood vessels as well as lymphatic vessels that basically bring all the nutrients, brings oxygen, brings carbon dioxide away, and then drains all the metabolic waste such as bile acids. So basically we're making an ecosystem both outside of the body, and we're transplanting into the body to understand how these organoids mature. And eventually our plan is obviously to see how we can use that to replace liver function, so a therapeutic approach. So that's a little bit of a peek of the sort of research that we're doing in the lab. But given that this event's theme is partnerships, I'm just going to talk a little bit about the variety of experts that we work together with and network together with to progress our research further. So historically, very briefly, our group first came about through the efforts of reconstructive surgeons at St. Vincent's Hospital, first Bernie O'Brien and more recently Professor Wayne Morrison. And back in the day, the term microsurgery, where you use microscopes to perform incredibly delicate, fine surgery on small blood vessels as well as nerves, was defined in Melbourne, mostly at St. Vincent's Hospital. And in the early days, researchers focused on how do you transplant back amputated limbs, amputated thumbs, and then when you have a patient with a large cancer that you take out and leave a large cavity, how do you reconstruct that? And the idea was to take bits of tissues from one part of the body and then transfer it to another to plug that gap. Very soon, after many successes, Wayne Morrison latched onto this idea of tissue engineering, where instead of taking one part of your body to replace another, why not grow a piece of tissue outside of your body in the lab? So creating something out of very little and then transplanting it back into the body to treat any sort of disease. So with tissue engineering in focus, the group started having a lot of scientists coming in. So Geraldine Mitchell, who's a vascular biologist, was brought in, who was interested in the manipulation of blood vessels, which is important when you transfer tissues around the body because it ensures that they survive. And a lot of other different scientists came in, and the research shifted from a very practical surgery-based research to much more scientifically driven research that focused on issues, for example, developmental biology, disease processes, as well as regenerative medicine. And then I came along over 10 years ago as a bright-eyed medical student, and I was entrapped by the idea that you could actually use stem cells to grow new human tissue outside of the body and actually put it back into the body. So I was incredibly excited by it, and I haven't really stopped since. I've been working on this ever since. But I think one of the main special things that I've noticed in my time is that research now no longer consists of a camp of clinicians and surgeons or a camp of scientists, but there's this free intermingling of ideas and cultures, and it's a really special place to be, especially for someone who can bridge both sides and go into both the clinician side as well as the scientist side. But I think going back to partnerships, the main thing is that there is no such thing as a one-person research or indeed even a one-team research. And a lot of research is driven by the network of teams that drive towards a common goal. And I think the more friends you have, the better you are at answering the big questions, and that's certainly been the case for us. So, for example, in our liver organoid research, we're experts in liver, we're experts in tissue engineering organoids, or we're experts in microsurgery. But the more complex our research is getting, for example, in making complex liver organoids, because the liver, after all, doesn't consist of just one cell type, but multiple cell types. And you can never be the master of all. So you need to have experts in each of the different domains of both assembly, different cell types, how you analyze them, and then how you drive that towards clinical output. So you need to have domains in each of those fields that work together cohesively. So in our case, we've got other experts, for example, liver cell biologists. We've got a group that we've just started collaborating with that works specifically on fatty liver disease. We've got groups that work on different cell types in the liver. We're always interested in recruiting more. We've got other groups that are specialists in stem cell biologists, particularly iPSC, as well as how to genetically manipulate them. And I also work together with other clinicians who treat liver disease as well as do surgery on the liver. And I think through Christine Wells, we've been exposed to this new sophisticated technology of both transcriptomics and proteomics, as well as metabolomics, which are very high-end molecular technologies that enable us to look at how cells behave and function at a level that we would never have been thought possible. And I think this just goes to show that when you're on the lookout for more opportunities and you're opening doors and you're talking to people and you've got experts who have fresh ideas, you're always opening doors to new opportunities that you never thought possible. And it could just be a simple case of someone sending me a paper and saying, I read this and I thought of you, and then it might be a new discussion, but it could be something much more important, for example, in Christine's case, bioinformatics, which I think as a clinician, what you often get is a readout of a patient's results, and then you make a decision based on that, but you don't understand how has that analysis taken place, why does that result come in place, and what can you do about it? You often have this robotic way of thinking in current clinical medicine where you're very protocol-driven, so your options of how to diagnose someone and treat someone is based on a menu, and that's based on a lot of research, but research done by other people. But I think a scientific mind is a curious mind, so as a clinician now doing research, when I see a patient, when I see a scan, when I see the blood results, I'm always asking questions. And I know that experiments and research is possible in any way. You just basically have to come up with an appropriate question and a research plan. And it benefits me that I've got scientists in place that can actually ground me in the realities of what's possible and what's not. So I think it makes me a better clinician as well. And I think that sort of mindset is the most important in driving the next generation of cures, both for diseases that are hard to treat right now, as well as those that are rare and underdiagnosed, as well as coming up with a new set of innovative solutions for things never thought possible 10 years ago, such as organoid transplantations. So I'm all ears, so if you've got any research questions, issues, I'm all ears. So ask away. Thank you.
Okay,
sure.
So what we're going to do after each of we're going to hear from 3 different groups today, and after each of the groups, we're going to hear an opportunity for you to ask questions. So a little bit of a different format, but please put your hand up. We've got a couple of microphones available, but I wanted just to lead off by asking you just to elaborate a little bit more about where you're at. So you've talked a lot about the different types of stem cells that you're employing in your research and your organoid work. And I had a feeling that the organoids that you've made, you've put back into animals to vascularize. You know, but are you going to be able to put these into to people, and if so, when? So what's the next steps for you?
Sure. So we're looking at 2 sides of the spectrum. So the first side is how do we make better organoids that are more therapeutically valuable. So that's one side, that's optimizing better organoids. And second is we're looking at how do we transplant these, so the surgical side, so the actual treatment side. And a lot of the surgical side is currently done on animals, but the models and the techniques that we're based on have been based on decades of reconstructive surgery work. And there's been clinical trials on the methods being done. And some of the transplantation methods have been done in small-scale clinical trials. So we're working on both sides and moving towards this divergence where it becomes more and more clinically relevant. But just going back to your point about how close are we towards actually transplanting it, we're a lot closer than we would have been 5 years ago because we are using human cells and we're using humanized models of animal disease. So animals that have a human gene inserted in them, animals that have a mutation that's found in human liver disease. So we're closer than we think, but there are obviously challenges in terms of making sure it's safe. And I think there's this caution towards overhyping how close you are towards actually transplanting things into a patient. But I'm reasonably hopeful. We don't necessarily focus, our group is focused on the practical aspects of liver organoids, but I am aware there's a lot of research on making sure iPSCs are safe for transplant. And so we're very much guided by those studies, but I always remain hopeful. Thank you.
Thank you. Are there any other questions? Yes.
Considering the livers are really big organ, how, like, are you going to grow it to the right size? How will you fix that?
So that's a very interesting question. I'm actually writing a grant on this tonight. So what we know is there's obviously different types of liver disease. So you've got liver disease where you have liver failure because of a drug response or because there's too much fat, and there's liver cancer, for example. And those, you actually need to replace a lot of the liver mass because the liver is approximately 5 kilograms in adult humans, and that's a lot of tissue with a lot of functions. There's about 500 different functions. So you need to replace a lot of that, and that's a very big challenge. And I think we're quite near that yet. But I think one of the things that we are near is towards patients with metabolic liver disease. And so that's a genetic disease where you might have a mutation in one liver enzyme, and because of that, there's a cascade of events that causes liver injury and subsequently disease. What we know from a lot of animal studies as well as preclinical trials is that by replacing 5% of that specific enzyme's function, so out of the 500 different functions, the liver has that one specific enzyme and that one specific function, and 5% of what a whole liver does, we're able to treat those patients. So I'm reasonably hopeful that that's probably our first candidate of patients that we would approach with these transplantations. Thank you.
So thank you very much. Please join me in thanking Kieran for his great presentation. I failed to introduce myself. I'm Megan Munty. Hello. And it's my pleasure to introduce our next panel. Please come on down. So joining us as Lincoln Stamp, Marlene Ho and Sebastian King. I'll allow them to introduce themselves, but Marlene, would you like to start? They're going to tell us a bit more about their work around stem cells and gut. Thank you.
Hi everyone. So on behalf of our team, I'd like to thank you, I'd like to highlight what a pleasure it is to be able to speak to everyone here today. And I'd like to thank the organisers, Christine, Negan, and also Trish and Helen for putting this all together. And, thank you. And, thanks Helen. And inviting us to speak. So just to introduce our team, Lincoln is here on the right, and in the middle is Sebastian King, and my name's Marlene. And so Lincoln and I run a small lab together here at Melbourne Uni, and we're interested in looking at the function of the gastrointestinal tract and also how to treat diseases of the gut. And to do that, we've partnered together with Baz, who is a paediatric surgeon at the Royal Children's Hospital. And working together with Baz has been really great, not just because he's a really friendly guy and brings a lot of energy into the room, but he's also given us some real insight into the challenges that his patients face and how we can address some of these issues with the research techniques that we're using. So as you guys all know, so the gut is the site where nutrients from all the food that you've intaken during the day is broken down and absorbed, and the waste products are accumulated and moved out. And so, as you can imagine, having your gut move in the right way is extremely important to your health. And I'm sure many of you guys have experienced that if you have any sort of upset in your stomach, or even worse, in your bowel, this is an extremely unpleasant situation, and it's not something that you want to continue through your life. So what's very interesting about the gut is that if you take out a piece of gut tissue, and this is an example from the large intestine of a mouse, what it's able to do is actually move independently of any sort of connections with the brain. This is able to happen because there's a nervous system within the wall of the gut, and it's responsible for controlling gut motility. So you've probably heard this being referred to as the second brain, and there's currently a lot of interest in how this nervous system in the gut talks to the microbiome, which is the bacteria that live within the wall of your gut, within the lumen of your gut, sorry, and also the brain in your head, and affects all sorts of things such as mood and also neuropsychiatric, oh my gosh, neuropsychiatric disorders. I probably didn't eat something very well today. So what's special about this brain in the gut is that they form this very extensive network along the entire length of your gut. So from your esophagus down to your stomach, and also throughout your small intestine all the way to the end of your large intestine. And just to give you a look at what these nerves look like, so if you open up the gut, you'll see many different layers of cells. There's epithelium, there's muscle cells, there's also blood vessels, there's immune cells, a big collection of different types of cells. And you'll find these nerve cells, which are clustered within these muscle regions. And Lincoln and I have been looking at these cells for the past 10 years, actually, and you can see that they're actually very beautiful. So these are some images from our lab. You can see the nerve cells in that form these clusters, which are called ganglia along the gut. The nerve cells here are shown in magenta, and they're surrounded by glial cells shown in green. And these nerve cells have these long processes that actually extend into the villi of the gut, and these are very important for nutrient absorption. So now, if you count up all these neurons in the gut, actually there's more neurons in the gut than there is in your spinal cord. That's why it's been called the second brain. But just to give it another plug, it's been shown in several studies that during evolution, the nervous system in your bowel probably developed before the nervous system in your head. So we actually like to think of this as not the second brain, but the first brain. Now, as you can imagine, because this nervous system in your bowel is so important for the control of gut motility, there's serious consequences if there's any issues during development, or if there's parts of this nervous system missing. And that's exactly what happens in Hirschsprung's disease. So this is a developmental disorder that affects about 1 in 5,000 human births. And what happens in Hirschsprung's disease is that there's segments of the gut where there's no nervous system present. And this can be a short segment at the very end of the colon, or it can be, in very severe cases, it can be no nervous system throughout the entire large intestine and extending all the way up into the small intestine. And what this means is that in this region of the gut where there's no nervous system, there's no control of gut motility, and actually there's no gut motility, so that the contents of the gut, accumulate, and that's shown here in this infant that's affected with Hirschsprung disease. You can see it forms as a megacolon, and this swelling of the gut is actually fatal if it's left untreated. So we use quite a few different, apart from the patients, which I'll leave to Baz to talk about, we use quite a number of transgenic mouse models and also a rat model. And you can see here an example of Hirschsprung's disease in a mouse with the megacolon, and so this is the small intestine, and you can see this progressive lack of neurons as you're moving along the gut. So I'll hand off here onto Baz to talk about what happens to these infants after they're diagnosed and the current treatment options available.
Thanks, Marlene. Along the theme of today, it's quite special to be able to collaborate with Marlene and Lincoln. So I'm a paediatric surgeon based at the Children's Hospital with a particular focus in colorectal conditions in children and congenital colorectal conditions and motility. And so for the children born with Hirschsprung's disease, the majority of the time this is the first member of the family to be affected. And so for those who've had children, babies born, you're expecting that they're going to be able to poo within the first 24 hours of life. But for a very small number, that's not the case. So about 25 to 30 children in Victoria each year will be affected by Hirschsprung's disease, about 80 to 100 around Australia. And so in that first 24 hours, when babies are normally expected to poo, these babies don't poo. And they progressively increase their abdominal girth over the next 24 to 48 hours, and usually come across to us at the Children's with grossly distended abdomen, unwell, with vomiting. And it's only at that stage that they get diagnosed with Hirschsprung's disease. And you can see there from the study on the left-hand side that actually what you've got is a contracted or spastic colon that is unable to relax. And the section above it is the grossly distended sigmoid or descending colon above it. And so those children, we need to get them out of trouble in the first 24, 48 hours of life by decompressing the bowel, getting rid of that gas and poo, and then over a number of months making sure that they're progressing well, that they're feeding, that they're growing, and eventually doing what we call a pull-through procedure, where I actually remove that section of bowel that is abnormal. And we do that through a combination of laparoscopy, so minimal access surgery, but also surgery via the anus. So we actually open up the bowel via the anus and pull that section of bowel through, remove the section of bowel that is aganglionic, and then take the ganglionated bowel, the section of bowel that has the nerves that we need, and attach that back towards the anus, whilst protecting the sphincters, and so hopefully being able to maintain the sphincter mechanism for the children and maintain their continence through life. Now that all sounds great, and a lot of the time it works very well, but unfortunately for a significant proportion of patients it doesn't work particularly well. They might have problems with chronic constipation, they might have problems with infection related to their constipation, a thing called enterocolitis, and that can actually be fatal. We've had one child die in Victoria this year related to enterocolitis, aged under 1. Or they can have problems with incontinence, and the incontinence might come from the fact that they've still got a bit of bowel there that works well, but it doesn't work as well as it should, or the sphincters don't work as well as they should. And that's where Lincoln and Marlene's work comes in, because how do we either do one of two things? How do we fix those children that have got problems post the surgery? Or actually what would really be really nice is how do we get rid of the requirement for surgeons completely whatsoever?
Thanks very much, Baz. That's not our ideal. We don't want to put you out of a job. But like Baz alluded to, we're actually really excited about trying to develop some alternative approaches to try and treat Hirschsprung's disease. And obviously stem cell therapy is something that we're really interested in. So our work in our lab and also with collaborators from the Royal Children's Hospital, including Baz, have shown that within your guts there is a neural stem cell population. And this even exists and is still present in the normal regions of a Hirschsprung's patient's bowel. And so the idea that we are trying to develop or approach is to isolate some of these stem cells from the bowel of patients, expand them or grow them up in dishes outside the body, and then ultimately transplant them back into that defective part of the bowel where the nerves are missing to make new nerves, and ultimately to try and restore function to that defective spastic part of the bowel that Baz and Marlene have spoken about. In the lab, we have mouse models that we've been using, and we've been able to show that when we transplant these stem cells into the bowel, these stem cells are able to proliferate, that is, they give rise to more stem cells, and they also generate more new neurons in the bowel, so they're making new nerves, and these nerves are able to function, and we've been able to show that they're incorporating or connecting up into the circuitry of the bowel. That is, they're talking to each other, but also importantly, they're talking to the gut muscle cells. And this is important to restore that motility that Baz and Marlene have spoken to you about. So our research program is moving forward to try and really model or incorporate the whole series of approaches and procedures that will be required to translate this stem cell approach for Hirschsprung's disease into a clinical setting. So one of the first key things that will probably have to happen in the clinic will be a surgical intervention to try and bypass this obstruction which causes the fatality in these kids. And we have a rat model of Hirschsprung disease where we're able to investigate this and then also ultimately test the ability of these stem cells to restore motility in the bowel. And this is where our partnership with Baz is really, really exciting and really important. So our Baz allows us access to human Hirschsprung's patient samples, where we're able to isolate some of these neural stem cells following the surgical procedure that he just spoke to you about. And of course this happens with all the appropriate ethical consents and parental consent forms as well. And this allows us to then test the potential of these patient-derived stem cells in our rat model of Hirschsprung disease and their ability to restore function into that part of the bowel. But moreover, our collaboration and connection and partnership with BAS is really important because BAS brings an extensive wealth of experience and knowledge, as Marlene said, to the needs and complications that these patients have after surgery, and this is really informing us on the future directions of our research program going forward. So, thank you.
So thanks, Lincoln, Marlene and Baz. So we've got time for some more questions, and I think I just wanted to follow on from the comment you just made around getting material from the families. So it's a similar thing, isn't it? You're using iPSCs. You're making iPSCs from that tissue, and then making them into nerves, and then testing them in your rat model.
In your guts, there are stem cells that reside within the gut wall. These are the same cells that made those nerve cells in the bowel during development. And we've shown that in rodent models, but also in humans, that these nerve stem cells are still resident in the bowel, and even in the small intestines or normal regions of the Hirschsprung's patient's bowel. So the idea would be to go up into the small intestine of the Hirschsprung's patient, take some biopsies from which we could isolate some of these neural stem cells, and then expand those cells as a neural stem cell population in a dish, and then ultimately transplant them back, so they'll be an autologous or patient-derived stem cell approach.
So the sample that you're removing at surgery that's defective, that doesn't have the nerve cells in it, but you can get biopsies from up higher to find the nerves.
So when we do what we call a pull-through for Hirschsprung's disease, there's a area of aganglionosis, which is a variable length. There's what we call a
Which means?
So it could be in the short segment disease that Marlene mentioned previously, might be 15, 20 centimetres. The patient that I did a pull-through on last week had about 50 centimetres of colon that we removed, so that was a longer segment. And then, as Lincoln alluded to, there are some patients that have total colonic disease where the entire colon is aganglionic, and then a very small percentage will have disease that goes even further into the small bowel. But the majority of the time, the 80 to 85% of the patients will have short segment disease. So we need to remove the area that is aganglionic. Then there's a transition zone as it goes from aganglionic to ganglionated, which is about 5 to 7 centimetres in most patients. And then it goes into ganglionated bowel. And so when we are either forming a stoma to get the kids out of trouble in the first place, or we're doing the pull-through, we make sure that we have gone back into ganglionated bowel, and we do what's called a donut, where we actually, rather than just taking a small number of biopsies, we actually take the entire circumference and do a frozen section on that, so that histologically we can be certain that there are ganglionated cells all the way around the whole length of the bowel.
Your first brain. Fascinating, isn't it? Any other questions? Chris?
So I was fascinated that even though you're working with human tissue, and I guess this is a question for Kira as well, animal models are still really important to understanding the disease. Could you comment on that a bit further?
Like Lincoln said, the project that we're trying to do at the moment is trying to mimic all the different steps that we have to do all the steps we have to perform in a human patient. But in order to do that, we need to be able to do that in an animal model and make sure that, first of all, the first step is already taken care of, so the pull-through surgery, and that's what Lincoln's now doing, sort of a reverse translation. We're curing Hirschsprung's disease in rats before we can do that in humans. So that allows the rat to be able to actually live in order for us to take the stem cells, expand them in culture, and then transplant them back and allow them to then grow in the gut and be able to innervate the muscle cells and talk to the muscle cells and have sort of gut motility, which takes time just within the one animal. So we need to be able to show that that procedure actually works in an animal model before we can get anywhere near putting it in humans.
So please join me in thanking our second panel. So I should mention that, of course, all the speakers will be joining us at the conclusion of the proceedings today and can answer more questions outside. So could I now introduce the third panel, Professor Elise Pibet, Dr. Matt Rutter, and Mr. Daniel Feller. Daniel, if I could invite you to join us at the microphone first, because I think you're going to lead and have a very important story to tell. So Daniel's from Genetic Cures Australia. Thank you, Daniel.
Thank you. Good afternoon, everybody. I'm just casually going to talk to you for a few moments about partnerships and how important these have been to our story. My son, Harry, is 8 years old, and he was born in 2011. As we soon were to discover, he has a condition, a genetic condition called Usher syndrome, which historically was termed deaf blindness. And as you can imagine, that's a pretty devastating diagnosis to be given for a child. We knew that Harry was deaf when he was 7 days old, but we didn't know about the Usher syndrome, his genetic disease, until he was 3 years old. So basically, let me talk to you a little bit about Usher syndrome. There are many forms of Usher syndrome, termed Usher syndrome type 1, type 2, and type 3. My son has Usher syndrome type 1, which means you're profoundly deaf at birth, and you gradually lose your sight in your second, possibly third decade. The Usher syndrome 2 is much more common, and that means that you have poor hearing at birth, which gradually deteriorates, and along with your eyesight, again in your second to third decade. So we have two other children. We have Tess, we have Alice, and we have Harry. Tess and Alice were not affected at all, and it was therefore a tremendous shock to be told that our third child had this thing called Usher syndrome. But very quickly after the diagnosis, which we had in 2014, it became very clear to us that through partnerships, and for me today is all about the partnerships that we've established, and talking about the partnerships through science, through government, through international advocacy for research into Usher syndrome, that we were not alone. We were far from alone, all being that Usher syndrome itself and the form of Usher syndrome that my son has is exceedingly rare, and we're not 100% sure. There's either 7 people who have Usher syndrome type 1F, which is the gene that affects my son in Australia, or there's 1. We're not too sure about that, but it's exceedingly rare. But the principles behind it are not rare. Deafness is caused genetically, blindness is caused through retinitis pigmentosa, which, as I said to you, becomes affecting in the second to third decade, and it's a gradual decrease of sight from the outside in due to retinitis pigmentosa. After the terrible diagnosis that we were given, we were introduced to Alice Pibe from the Centre for Eye Research, to Alex Hewitt, to Megan, to many others, and it became very clear very quickly that through partnerships with teams around Australia, the outlook to Harry's diagnosis was far from awful. I was given the privilege of speaking at the ISSCR conference last year, and in that we spoke about how stem cell research meant the future for Harry was so bright, and I managed to be able to say at one point that Harry's story is far from appalling. I don't know if you remember this, Megan, but the crux of my speech was Harry's story is far from appalling. In fact, it's a beautiful story. It represents partnership of medical teams throughout Australia, throughout America, throughout France and Europe, who are all coming together and working in their own ways. And as Kiryu said, there are no silos. Scientists are working together, albeit doing separate work. There are no silos. And it's very heartwarming to see that all of our partners are working towards a common goal of the use of stem cell research to cure these incurable diseases. And that is exactly where I come in. I am no scientist, I can assure you of that. I'm a concerned father, and my role has been to advocate the use of stem cell research, the exciting opportunities that offers us, but taking that to politicians, taking that to business, pushing for advocacy, pushing for funding to research, for ongoing research, which is what I do. So I have approached government through the charity that we set up with Alice Pibe, with Alex Hewitt, with the University of Melbourne, called Genetic Cures Australia. And through Genetic Cures Australia, we have approached government, we have approached business, and we have made enormous inroads into research for this condition and others. The Health Minister has been very supportive of us, but his prerequisite was, your son is one. Your son is one person who has Usher syndrome, which is a genetic condition which can be potentially treated through stem cell research. If we were to help you with this, would that one treatment then be able to be cut and be pasted to other forms of inherited blindness? And I certainly wasn't going to say no, that's not the case, but the truth is, of course it is. Once one thing can happen, then the treatments can be cut and pasted to a whole host of diseases, be it inherited blindness, be it motor neurone disease, be it anything else that affects the body. That is the theory. And if my son is one of the first to benefit from this, then nobody's going to be happier than me. But the whole point is, it's all about partnerships. It's all about bringing together businesses, bringing together government, bringing together medical researchers from all over. And this is what I've had the privilege of being able to do by advocating research for my son. So it's a bit of a random speech today, and I'd like to hand over to Alice to continue to focus on the work that we've done. But in essence, it has been an absolute privilege to bring together many facets of Australian society to help my boy. Thank you very much.
Thank you everyone for being here tonight. It's always really heartwarming to see people from the community come and listen to us. I always make the joke that as a biologist, we live in labs, we don't speak to people that often, we speak to cells, sometimes they answer, and we know it's time to take a holiday. So when we have people here, we're very, very excited because we want to share our work. I think it's very important that the community understand what we are doing, given that we aim for impact, we aim to make for a better world where we find cures for people to go on with a better life. So in my job as a stem cell scientist, if I can get that to work, will be the first step, is one of my main interests in my research is to try to model diseases of the eye. And I do this with a team of people, with collaborators, but what we use as a way to model diseases of the eye is a specific type of stem cells called iPSCs that were introduced earlier in the meeting today. An iPSC is an interesting cell. Basically, we can take today, and we do this routinely with my collaborator, Alex Hewitt, who is a clinician. We are able to take skin samples from any individual. We work on vision, so we take generally people with eye diseases. We take the skin cell and we turn it back in time into a stem cell. The word stem indicates that it can become everything. And with this, we ask those stem cells to become the cells of interest that we are interested in eye diseases. The reason we do this is because maybe we don't realize this, but when we look at blindness, there's a lot of conditions that affect vision, but we know very little of why vision is lost. In many conditions, including in age macular degeneration, we do not know which cell type dies first and why. The image I put at the top here is an image of a retina. So that's the back of your eye. That is the one part of your eye that, for those who see, is able to transform a wavelength, so light, into a signal that the brain will then interpret as an environment that is around us. If you take a piece of paper, which you all have today because we gave them to you before you came in, if you just look at the thickness of that paper, the neural retina is thinner than that thickness of paper. And it is made of different cell types that all play a role in vision. What is interesting about diseases of the eye is that we often don't have the right model to model the condition of a human eye. which is why most likely we don't understand really well why people lose vision, because we don't have the right model. And that goes back to my point of what I do as a living. I'm trying to make a biopsy of the back of the eye without taking a biopsy of the back of the eye. We bypass the biopsy by taking skin sample to make the back of the eye. And once we have this back of the eye in our plastic dish, we are then able to interrogate what goes wrong in the cells that are affected in people with those conditions. So in the case of Harry, this is, when we first met, we thought this is just a wonderful opportunity for all of us to work together and to try to make it better and try to understand a condition for which we know very little. But as you said, Daniel, what we do is, in theory, accessible to many other conditions. This is one genetic condition we talked about, Usher, but we can link it to many other conditions being much more widely observed in the population, such as, let's say, AMD or glaucoma. And this is really the work we do. We're trying to model disease in a dish for conditions for which we don't have good model in the current animal models we have. And I will now pass it on to our next speaker, who can complement the work we're doing together.
Thank you, Elise. So I'm coming from a very different angle, I guess, being an immunologist and a neuroimmunologist at that. But as Elise mentioned, I'm going to talk a little bit about some of the, put my slide up on my help, a little bit about some of the work involving a very valuable partnership we've forged, looking at exploring really ways of modeling inflammation in eye disease using stem cells. So just a little bit of background to that. So the immune system is strongly implicated in a large number of neurodegenerative diseases. And these include things like Alzheimer's disease in the brain, but also in the eye, obviously, which is a strong interest of mine, diseases like AMD, diseases like retinitis pigmentosa and others. And so it's extremely important then from that perspective to be able to have tools to better understand how these processes like inflammation work, and also to then in turn develop better treatments that are able to deal with these particular diseases. Now, unfortunately, diseases like AMD and Alzheimer's have a very strong lack of treatments. So, and while this work is very interesting, it presents quite a challenge because of the environment of the brain and the eye. So the brain has its own special immune system or its own special immune population. And these are these, these are these little microglial cells as shown in green here. And you can see they're highly ramified cells with very long, elaborate processes. And so what they do is they principally help maintain our nerve cells or maintain our neurons. But in the course of degeneration or neurodegenerative disease, they can actually spur powerful inflammatory responses which can actually exacerbate injury and exacerbate degeneration. And so these are actually very elaborate and very dynamic cells. So if a little video here, if I can play it, this is a tissue section from a mouse actually. In the center here we have a lesion which has been induced via laser. So if we play this time-lapse video of only a few minutes, will it actually allow me to do this? We can see, if we look towards the center, we can see the processes of these microglial cells are very dynamic, and they actually move towards and migrate within only a few minutes towards this lesion site. And so in the process of that, they then start activating these powerful inflammatory responses. And so this is, I guess, an example of the fact that it's highly desirable to be able to control the activity of these innate brain and eye immune cells as a means then to potentially suppress these damaging immune responses. And again, these responses are ones which lie at the heart of diseases like AMD and diseases like Alzheimer's. The problem that arises, which brings me to the partnership that we've developed, is that it's extraordinarily difficult to model these cells given that they occupy a very specialized niche within the brain and within the eye. So isolating those, and particularly isolating those from patients, presents a really real problem. And so through the collaboration with Elise, my main slide has been already shown, but anyway. Surprise as well, but anyway. So through that collaboration with Elise, we've been able to develop a very sophisticated system for modelling this sort of neuroimmune system or this microglial system in a dish. And this is utilising the stem cells, and like Elise teased in the previous talk, utilising stem cell technology to create these sort of mini retinas or mini eyes. And so from the work we were doing, we're not only making those mini retinas, but we're actually making microglial cells from stem cells from patients, and then integrating those together to create this sort of immune environment. So these are fluorescently labelled microglial cells here. And so using this very unique approach, this grants us not only the ability to advance first our knowledge of inflammation and how it impacts retinal disease and how these very important brain and eye immune cells are involved, but it also allows us to work towards our sort of main goal, or our main goal to create a platform for more personalised screening that allows us to utilise that for patients and look at, very importantly, at inflammation targeting treatments, including for diseases like AMD, but it also has relevance to other diseases such as Alzheimer's and other eye diseases like retinitis pigmentosa, which we're hopefully going to push towards very soon.
Thanks, Matt. So again, time for questions. I think it's really interesting to hear about how complex these models are becoming. So I think one of the limitations with some of the early models, they were perhaps few cells, and of course our organs are very complex. There's a lot of interplay between different cell types. Matt, when I was looking at your image, even the preview of the image, I was thinking about the challenge of trying to appropriately represent the different cell types. So how do you kind of manage to get the right number? Do you know what the right number of immune cells are? Like, how do you?
That's a really good point. A lot of our time, is spent trying to benchmark and showing or to demonstrate that I've got the right cell in terms of what markers it's expressing and functionally how it's working. But you're right, these cells not only have a particular set of markers or particular phenotype, they also need to spatially occupy an appropriate sort of space. And as shown in that image to the left, these microglial cells have a real tiling pattern which needs to be recapitulated. So while this image on the right looks seemingly chaotic, it's also depicting a developmental process that's happening. So these cells are actually still migrating and working their way into their environment. But one of the ways that we are able to benchmark and determine that they're developing appropriately is that we utilize techniques such as live cell imaging here, where we're actually able to look as these little mini eyes develop and seeing how these cells become organized and hopefully how they then incorporate an accurate niche of what we hope they will become.
Well, thank you. Elise, just following on from Matt's point around maturation, and I think we heard a little bit about that from your talk as well in terms of vascularization and maturation of cell types. I mean, how immature are the organoids? So Matt used the word developing, but do they represent a mini eye or mini retina? Or?
Yeah, and I guess that's a very exciting thing in stem cell research today, which is that we are able to grow what we call mini organs. We call them organoids, because they do recapitulate human development of different type of organs. So we can make them for the heart, we can make them from the heart, we can make them for the liver, we can make them for the eye and the retina in particular. Because they follow development, that means that they're not like a fully mature organ that will be in me, for instance, but they do follow the first few months of development of a human. So they're not fully mature, but that might be okay. If you're looking at a genetic disease that affects a young individual in development, you actually want something that is quite immature to look at the first event in development that go wrong. Why? Because if you can look and understand what goes wrong very early, then it becomes a very early point of intervention.
Oh, very good, thank you. Now, Daniel, I'd like to give you the final word before we hand back over to Christine. You mentioned international partnerships, and I think we haven't really had a chance much to tease that out in terms of the scientific international partnerships, but I just wondered whether you'd like to elaborate a little bit more about the value not only attending the International Society meeting in terms of a scientific community, but that broader network that you've established with the parents around the world, but also the scientists and clinicians? Because I think your network is pretty extensive.
We have done this. We've spent 5 years now getting to know teams, other Usher syndrome societies, particularly on the east coast of America, but also England and elsewhere. But I think I can sum up everything by just a very small anecdote. We had an Usher syndrome conference for exactly what Harry has in Vancouver a couple of months ago. And all teams around the world, of which there's a few now, who are doing work, presented. And I was told that our team's presentation was fabulous before we went by someone who'd seen it. And I specifically decided not to ask, because I wanted to see it and hear it at the event in Vancouver. When our team stood up and spoke for 15 minutes, they spoke about problems. Because in the time that this person who had heard the speech, 3 weeks later, things had developed so much in those 3 weeks that it was no longer relevant to make that speech. It was relevant to make the speech of what had happened in the 3 weeks since. Now, this alone reflects on the speed of change. So our team presented a problem that they'd had with CRISPR, with base editing. As I say, I'm no scientist. Forgive me, it's Mandarin to me. They presented problems that they'd had, but because of who was in the room, those problems that our team were having were solved within 24 hours. And that would not have happened had it not been for advocacy around the world, for everyone talking to everyone. And as the previous team was saying, there are no silos here. Everyone's working together. There's no playground politics about who's going to be the first, and that I can attest to. So, had none of this happened, we'd be in the same position that we were in for 3 months, which we can't have. We're on a timeline here. But thanks to that, thanks to everybody working in our little area of the playground, this was solved, along with the other 20 things that were solved because of that conference. So it's just a small example, but it's a vitally important example that everyone is working together towards the same goal. So in answer to that, that would never have happened had we not had the teams from all over the world, quite frankly.
Oh, thank you for sharing that. And I'd just like to say that I think your Mandarin's very good. Please join me in thanking the team. And I'll just hand over now to Christine.
Thank you very much, Megan. Thank you, Daniel, Elise, Matt, Lincoln, Marlene, Sebastian, and of course Kiru, for giving us a snapshot of your worlds. I'm very proud that the University of Melbourne Centre for Stem Cell System has over 50 research groups who work collaboratively to tackle a number of different problems with stem cell research and regenerative medicine. These scientists include mathematicians and statisticians, material engineers, legal scholars, ethicists, and it takes that village to really tackle the genuine translation aspects of stem cell research. I think the other take-home message from today for me is that there's not just one type of stem cell. There's no magic bullet here. It actually requires a number of components. The first is to really understand the tissue or the organ that you need to treat. The second is to really understand which cell is going to be the cell that provides you with a property that you can study or address. And the third is that you've got the right people in the room, the partnerships, which are not just the ivory tower academics sitting locked away in a laboratory behind the security screens, but actually that we're building these partnerships back out with our clinical colleagues and with our community partners to ensure that the quality of the questions we are asking and the urgency of the questions that we are asking are relevant and timely. And I think this really reflects the future of stem cell research from my perspective. So we have a website where I invite you to come and explore the other stem cell research that is happening on campus. There are a number of stem cell colleagues here in the audience. Please stay, talk to them, talk to our panellists, enjoy a drink and something to eat, and get to know us a little bit more. If you have any other questions for us, you can certainly contact us through the email addresses on the website or with the contact information in the handouts that you've been given. Thank you very much for coming along. We've really appreciated your attendance. Thank you to our panel as well.